Welcome to the deep dive. Have you ever stopped to think what's truly powering, well, everything in your body? Digesting food, thinking, moving, often as proteins. We hear about them all the time, right? Diet, supplements, the building blocks of life. But let's get past the headlines. What are these things, really? Today, we're diving deep into that, using the sources you sent us. Our mission, to unpack the secrets of proteins from their tiny parts to their huge jobs. Get ready for some aha moments.
Indeed. And we'll look at their, complex structures, their vital roles, and even some surprising properties that scientists use in the lab. It's really a journey from micro to macro.
Okay, let's start right at the beginning then. Proteins are huge, complex molecules, we get that. But what are they made of? Like the absolute smallest pieces.
Right. So at their core, proteins are what we call polymers. Think of a very, very long chain, like beads on a string. These chains are built from smaller units. For proteins, these units are called amino acids. They really are the fundamental building blocks, like you said, like tiny bricks making a huge wall.
Bricks in a wall. Okay, I like that. So, how do these bricks connect? Is it always the same way?
It is a specific connection, yes. It's a chemical bond called a peptide bond. And how it forms is interesting. It's a condensation reaction. Basically, when two amino acids link up, a water molecule is released. Squeezed out, you could say. That's the condensation part. It makes a really stable link between them.
Huh, squeezing out water to connect. That's specific. And these chains, do they have like a start and an end? Does it matter which way you read them?
Oh, absolutely. Directionality is key. One end always has what's called an N terminal. That's a free amino group. The other end has a C terminal, a free carboxyl group. And here's the important bit. New amino acids are always added from the N terminal towards the C terminal. It's like a one-way street for building the protein correctly.
Got it. A molecular assembly line. And do we have names for really short chains, like just two or three of these bricks?
We do, yeah. Two linked amino acids make a dipeptide, three make a tripeptide. Proteins, though, can be, hundreds, even thousands long.
Okay, so they're built from amino acids. How many different kinds of amino acids are there? What makes one different from another? They can't all be identical bricks.
Definitely not identical. Good point. Each amino acid has a central carbon atom, kind of like an anchor. Attached to that are an amino group, a carboxyl group, and a hydrogen atom. Those are common to all. But then there's the fourth thing attached, the R group, or side chain. That's the unique part. It determines the amino acid's specific properties, its personality, if you will. And there are 20 common types, each with a different R group. And something else really interesting, most amino acids have a handedness, like your left and right hand are mirror images. It's because of how those four groups are arranged around that central carbon. It creates what's called a chiral center, and crucially, in nature, proteins almost exclusively use the left-handed versions, the L isomers.
Left-handed molecules. That's wild. So our bodies are built with just one type of hand. Unlike sugars, right? Don't they use the other hand?
Exactly. Sugars mostly use the D isomers, the right-handed ones, the fundamental difference.
Any exceptions to that handedness rule in amino acids?
Yes, one key exception, glycine. It's the smallest one, and its R group is just another hydrogen atom. So it has two identical groups attached to that central carbon. That means it's not chiral, no handedness for glycine. It's unique that way. And, when we talk about them, scientists use different names. There's a full name like glycine, the three-letter code GLI, and really useful, the one-letter code G. These single letters are super handy for long sequences, especially in bioinformatics for computers. Some are obvious, A for alanine, but some you just have to learn, like W for tryptophan, Y for tyrosine, K for lysine. It's like a little molecular alphabet.
Okay, a whole new alphabet. So we've got this chain of different amino acids strung together. How does that become one of these complex machines doing jobs in the body? It can't just stay a floppy string, can it?
No way. It absolutely does not stay a floppy string. That linear sequence is just the start. This is where protein folding comes in, leading to the four levels of protein structure. Proteins fold into very specific 3D shapes or conformations, and that final shape is absolutely critical for what the protein does. If the fold is wrong, the function is usually gone.
Okay, four levels. Let's break that down. What's level one?
Level one is the primary structure. It's simply that linear sequence of amino acids we just talked about. The order they're linked in from N terminal to C terminal, just the sequence itself.
Okay, the basic sequence. What's next?
Next is secondary structure. As the chain gets longer, parts of it start forming regular repeating shapes. The two most common are the alpha helix, think of a spiral coil, and the beta pleated sheet. You can picture the beta sheet like like a pleated skirt kind of folded back and forth. These shapes are held together mainly by hydrogen bonds in the protein's backbone, not the R groups yet.
Spirals and sheets. Got it. Then level 3.
Tertiary structure. This is where the entire polypeptide chain folds up into its final complex 3D shape. It's the overall shape of one single protein chain. Now, the R groups really come into play. All sorts of interactions happen between them. Hydrogen bonds, ionic bonds, hydrophobic interactions, pulling oily bits together, and importantly, disulfide bonds.
The sulfide bonds, what are those?
They're strong covalent bonds that form specifically between sulfur-containing amino acids like cysteine. They act like molecular staples, really locking parts of the protein structure together.
Okay, so tertiary is the whole chain folded up. What could possibly be level 4?
Quaternary structure. This happens when a functional protein is actually made up of more than one polypeptide chain. Each chain folds into its tertiary structure, we call it a subunit then, and then multiple subunits assemble together. A classic example is hemoglobin, the protein that carries oxygen in your blood. It's made of 4 separate subunits, 2 called alpha chains, 2 called beta chains. They all have to fit together perfectly to work.
So it's like a team of proteins working together as one unit.
Exactly. It shows how proteins can collaborate to perform really complex tasks.
That complexity is mind-boggling. So, okay, they fold intricately, but what are they actually doing all day? Why should we care about all these bonds and folds?
The why care question. Because they do practically everything. Proteins are the absolute workhorses of the cell, of your entire body. Their diversity of function is just staggering. I mean, think about enzymes. Pretty much all enzymes, the catalysts speeding up chemical reactions in your body, are proteins. Without them, life would just grind to a halt.
So they're like little machines speeding things up. What else?
Well, they do storage. Think of albumin in egg whites. That's protein storing nutrients for the developing chick. They act as hormones. Insulin, which controls your blood sugar. That's a protein hormone, sending signals. They provide structure. Collagen gives your skin and bones strength. Keratin in your hair and nails. All structural protein.
Right, like the body's scaffolding.
Precisely. Then there's defense. Antibodies fighting off infections. Proteins. Transport, like we saw with hemoglobin carrying oxygen. Other proteins transport fats, vitamins, minerals, and receptors on your cells. They sit on the surface, catch signals from outside, and tell the cell what to do. Communication.
Wow. Okay. Enzymes, storage, hormones, structure, defense, transport, receptors. They really are involved in just about everything. It connects the chemistry to, well, us. So beyond structure and function, you mentioned they have some other interesting properties that scientists can use. How they change, maybe.
Yeah, some really fascinating properties. Take denaturation and renaturation. Denaturation is when a protein loses that specific 3D shape. Usually this means it loses its function too. The classic example is cooking an egg. Heat denatures the albumin protein in the white, turning it from clear liquid to solid white. And you can't really uncook it, right? That's often irreversible.
Right, no going back for a cooked egg.
But, and this is amazing, some proteins can refold. If you gently denature some proteins, maybe by changing the environment slightly, and then return the conditions to normal, they can sometimes spontaneously fold back into their correct functional shape. That's rematuration. Understanding this is huge because misfolded proteins that don't refold properly are linked to really serious diseases like Alzheimer's or Parkinson's. So that folding process is incredibly important and sometimes fragile.
Wow. Okay, so folding and unfolding is key. What else?
Another big one is their sensitivity to pH. Remember those amino and carboxyl groups and some R groups? They can gain or lose protons, hydrogen ions, depending on how acidic or basic the solution is. This changes their electrical charge. Proteins are covered in these changeable charges, making them very responsive to the pH of their surroundings.
So their charge changes with pH. Does that lead anywhere useful?
Oh, definitely. It leads to the concept of the isoelectric point, or pI. Every protein has a specific pH value where its overall net charge is exactly 0. The positive and negative charges on it balance out perfectly, and at this pI value, proteins are often least soluble. They tend to clump together and precipitate out of solution.
Like curdling milk.
Exactly like curdling milk. Changing the pH to the pI of milk proteins makes them clump. Scientists use this trick all the time in the lab to separate and purify specific proteins from mixtures. It's a really powerful technique.
Huh. Clev. What else can we do with them?
Well, some amino acids have a neat property, light absorption. Specifically, phenylalanine, tyrosine, and tryptophan have ring structures in their R groups that absorb ultraviolet light, particularly around 280 nanometers. So you can just shine UV light through a protein solution and measure how much is absorbed. It's a quick way to detect proteins and get an idea of how much is there without adding any dyes.
So they kind of have a built-in signal for UV light.
Pretty much, those three do. And there are chemical tests too. The ninhydrin reaction is a classic. You add ninhydrin solution to something, heat it up, and if amino acids are present, it turns this characteristic purple color. The deeper the purple, generally the more amino acids you have. It's a simple visual test used for decades.
So we can make them clump, see them with UV light, or even turn them purple. Lots of ways to study them. We have covered a lot today. We went from tiny amino acid building blocks linking up all the way to these huge folded machines with multiple parts like hemoglobin. We've seen how that intricate structure is everything for their function, how their chemistry lets us, actually study and work with them.
It really shows how much complexity is packed into every single protein. They truly are molecular marvels working nonstop inside you. Yeah, just think about it. Right now, countless proteins are digesting your food, carrying oxygen, fighting off germs, sending signals in your brain. Almost everything is orchestrated by these incredible molecules.
So next time you think about proteins, maybe eating them, maybe just learning about biology, take a second. What other things happening in your daily life right now might be powered by these amazing biological workhorses? What else are they up to?